Good morning, everyone. Thank you to everyone that's here to participate of our third webinar. So today we have the presence of Horacio Guzman, who is currently in Slovenia, but he is from Bolivia, and Danilo Gonzalez from Chile. So it's a really nice webinar because we have two words about SARS-CoV-2 is a huge problem that the humanity is facing nowadays. So first, I want to thank Horacio and Danilo for accepting the invitation to talk to us today. And I don't know if you want to say something before we start.
Oh, no. Good morning, everybody.
Good morning, everyone. Yeah.
Thank you, Rafael, for inviting us. That has been a great opportunity to show what we are doing in our labs, in my lab at least.
Yeah. Okay, so we will start with the talk from Horacio. So please share your screen.
Yeah. Okay.
Okay, so here he is. So Horacio will talk a little bit about some free energy calculations from the spike proteins of coronavirus. So Horacio, please, it's all yours.
So thank you, Rafael, for the introduction, and thank you very much for the invitation to the whole SAIMS organization and the seminar. Well, today I'm going to talk a bit about the coronavirus, the spike proteins specifically, and the free energy characterization we have been doing. Everything has been done with this multi-scale perspective or under these multi-scale methods, which is meaning that we have at least molecular dynamics in all atoms, and at the same time, we have a molecular dynamics coarse grain, and or some continuum method related. So I'm going to start with a basic introduction and the motivation on why are these computer simulations fundamental sometimes. And again, if we see that sometimes in the real experimental life, when experiments are done, for example, in the case of drug design, which is, I think, the topic Danilo is also going to tackle in the next talk, then we basically some of the experiments done are very expensive and also very time consuming. And that's why using computational models for such drug design or a code design helps a lot, like virtual screening, among other techniques. On the other hand, also when experiments are blind, this is the case, for example, of the atomic force microscope. When it's operating in the dynamic mode, we know that there is a force measured between a surface and the tip, which is oscillating. And we know we can the tip can sense the whole force of the interaction, the total intermolecular force, but not distinguish where is the physical origin or which is the physical origin of that total force. So in those cases, also the computational modeling are necessary, because we need to observe different time scales that are not always reachable by experiments. And in particular, for example, in the nanometer scale, where we are tackling the viruses. For example, it will go from a couple of hundred of nanometers to, of course, the Armstrong level. And just a short reference, in case many students are watching this seminar, it's really good to look back a bit in the history to see where we are now. The concept of microscopy was at least in it was at least found at the beginning of the, or not the beginning, the end of the 17th century, so 1677, by Van Leeuwenhoek, who was observing under a concave glass against the light some microorganisms. However, the start of the computational microscopy happened just in 2000, around 2015, or in these decades of the in the year 2000. I would say in the 2000th century. But this was, of course, a work that was carried out, and a term that was carried out by Klaus Schulten, where he actually indeed simulated a whole viral capsid with all atom simulations in, I think it was in the blue waters. It's a huge supercomputer. And again, so the scales where we are located are of course multi-scale, because these proteins that are comprised within the virus capsids work at different time scales. For example, let's say from vibration of bonds from a few femtoseconds, picoseconds, going to the protein diffusion, which will be at a couple of milliseconds, and other phenomena going on in these biological huge ensembles. On the other hand, from the length scales, we can see that we have also at least 3 orders or 4 orders of magnitude if we want to look at viral capsids. And all these orders of magnitude make the simulations computationally very heavy. But just a short introduction now on what is virus. I think due to the pandemics, everybody now has a very good idea. Well, in a look from a physicist, it will be basically comprised by the viral capsids or envelope. In this case, if we're talking about SARS-CoV-2, it's an envelope. And inside we have a genetic material, and then we have the glycoproteins or spikes, which are mostly doing the recognition process with cell enzymes. About the virus forms, they are well classified. For example, icosahedral virus, conical virus, and even some other asymmetric virus are kind of classified. And we have even in our rule the T number, which show us exactly kind of T geometry has an icosahedral virus. On the hand of modeling virus capsids, we have, for example, the Fokker von Karman numbers, which allows us to study the form of a viral capsid with analytical theory at the continuum level. And this analytical theory is based on this relation, which basically shows that delta R, which is the swelling that can occur at the capsid, in reference to a reference radius of these capsids, and the swelling relation to the pressure and to a macroscopic, let's say, Young modulus involved on this swelling mechanism. And of course, given a certain Fokker von Karman number, we can study analytically how the swelling how the swelling of icosahedral capsid behaves as a function, for example, of the exerted pressure, and also how the sphericity radius of this capsid works. On the other hand, we can also see the internal, mostly the internal pressure inside the capsid analytically. In this case, is for icosahedral capsid T3. It's here in the red circles, and in the analytical case of a sphere, it's in the dashed line shown here. This work has been done also quite recently, and can be extended to look at some environment mediated variables, for example, the concentration of ions. So the ion strength is shown here at the right side until this equation of the pressure according to the Weierhuckel regime of those icosahedral capsids. And which is the restriction of this analytical theory, which actually show us good guidelines of what is going on, and regarding in which regime of if we are in the regime of the Weierhuckel, we can see the pressures here and how they behave as a function of the concentration of ions. However, they are not considering the molecular details. The molecular details is what we want to see normally via computational models like molecular dynamics. And in molecular dynamic simulations, I think most of you know that we define total energy based on different interactions, bonded and non-bonded. As bonded, we have bonded interactions, angles, dihedrals, and non-bonded, we can have typically some electrostatics or Lennard-Jones type potential, for example. And then we solve the Newton's equation with the energy term here. And that's basically the starting point from molecular dynamics. Now I'm going to talk about the method, these multi-scale methods of free energy characterization that is the main topic of the talk today. And I'm going to do that by introducing a work we done with, not with SARS-CoV, we have done this work with the tratoma virus. And actually, it was work done by our student from Chile, and it's a collaboration with the University Santa Maria of Valparaiso. And this work is showing exactly what is our aim in terms of multi-scale methodology for free energy calculations. So this work has the following premises. We have an hypothesis to go beyond the proof of concept of the modeling. So if we are talking about, in this case, a multi-scale model, we don't want to be restricted to just the smallest protein we can find on the PDB. We wanted the method to be ready to tackle huge viral capsids and other biomolecular assemblies. And from the side of the observables, we wanted to, of course, the method to be quantitative and go as far as possible along with the latest experiments. And experiments in both, let's say, with high resolution, molecular resolution, atomistic resolution, and static or dynamic experimental methods. So the idea of behind these multi-scale models are that we can basically go from an atomistic representation to let's say a coarse-grained representation by simplifying some degrees of freedom of the atomistic representation. Of course, it doesn't mean that the whole properties and the whole physics are going to remain the same, but we can, of course, target some properties that we are interested in and build such coarse-grained models. The transferability between models is normally a very, very difficult topic because it doesn't happen all the time. But as I said before, for many properties in the model, we can basically go, for example, if we want to see the structure, the atomistic structure, there are ways to achieve a back mapping strategy from a coarse-grained simulation. And these back mapping strategies are normally achieved by doing some also small relaxations at the all atom level. However, this is the main concept we have. We are in this case multi-scaling length scales. So we are not using any enhanced sampling methods. So the method I'm going to present, it's basically multi-scale in the sense of lengths. Well, the method has also been published in this special issue of the SAIMS that was related to the JSON. So we were ready with the methodology to do a study of huge macromolecular protein assemblies, for example, of various capsids. However, we didn't find at that time, I'm talking about the last quarter of 2018, we couldn't find molecular trajectories, none from experiments and none from molecular simulations of the disassembly of virus capsid. And then suddenly one of our collaborators suggested to look at an article which is cited here below, where they used a coarse-grained strategy, which is based on the CIRA method. And the CIRA method allows multi-scale also modeling. For example, in the case of water, they have different levels of water. They have the atomistic water. They have this WT4, Wat4, and what else levels of coarse graining in water, which will nicely build, as we see here from the density profiles, within the same simulation box, multiple length scales of water molecules that are basically connected together. And also there are some rules for different kind of for all proteins. And through this methodology, we found some molecular trajectories showing the electrostatic repulsion effect of some negative ions, negative charges inside the tritoma virus. And when we see this publication, we were saying, okay, this is perfect to test our method. So what we did is basically from the atomistic capsid structure of the tritoma virus, we were in coarse graining, and then we started this dissociation of the capsomers by adding in the middle inside the capsid a lot of negative charges, and thus starting a strong electrostatic repulsion. So with this electrostatic repulsion, we see that which are the disassembly paths of this capsid, and we just apply the backmapping procedure, do some relaxation, and go to a Poisson-Boltzmann solver. So now we have changed completely scales from a coarse graining representation, which gave us at the end, through the backmapping, atomistic resolution of the whole capsid. And this atomistic resolution now is used inside a Poisson-Boltzmann solver. So we see here the boundary conditions. We have, in this case, 3 regions: the regions inside the capsid, outside the capsid, and the capsid itself. And we tackle basically 3 cases where the capsid is assembled without negative charges inside, where the capsid is assembled with negative charges inside, and the case C, where the capsid is already under disassembly, starting the disassembly with the negative charges inside. And we could see, and we could prove that the mechanism of the electrostatic repulsion makes sense from a free energy viewpoint. And this is a very, let's say, efficient method, because to doing so, we just needed after the coarse graining of the viral capsid and the backmapping, we needed just a couple of hours to calculate the free energy through Poisson-Boltzmann of the whole capsid. And we see, in fact, that, of course, the case C, where the capsid is starting disassembly, and we have ions inside, it's the most favorable in this case compared to the one where the capsid is completely assembled. So passing now to the SARS-CoV-2, well, this is an animation. We see the EC2, we see the 3 S proteins, and seems very fast. The nanomechanics, we first studied with collaborators in the US and in Poland. We studied the nanomechanical properties of the SARS-CoV-1 from year 2002. And we, just briefly, I'm summing up. If you want to see more details, you can look at the paper. We see by some pooling of the RBD domain, SARS-CoV-2 has more resistant, let's say, has higher mechanical properties than the SARS-CoV 2002, and as stable. So the other research we are performing from the side of COVID and SARS-CoV-2, it's to do the characteristic to the method I presented before with Poisson-Boltzmann calculations of the whole spike protein. So in this case, what we want to see is under different pH concentration of salt, the salinity, by varying the salinity and the pH of the environment, how is the behavior of the spike proteins? And why we want to do so, and why this project started, because for SARS-CoV-2, they found basically the virus in many different organs, and we want to understand exactly where is it feeling comfortable, or where we may find some weaknesses. And then we started project, which is called Pi for All. And we can see that in Pi for All, we could find different trajectories, molecular dynamic trajectories of, for example, the spike protein, as was given even by Dee Shaw or many groups in the US. Also in Europe, they were sharing it through the MolSI or BioExcel and many other platforms. And through these trajectories, we can basically do a rapid free energy characterization with our method, with Poisson-Boltzmann solver, and then proceed, of course, with the quantitative analysis as a function of the pH or as a function of the salinity and so on. And for this, what we provided is, and the project has already a GitHub page. The API is not launched yet, but we are about to launch the API. What we provide is not only the speed of code in terms of the molecular dynamic engine that we are using, which is C or C++. The speed of development, which most likely must be Python or something like this to be rapid, and the interactive shell, but also we want to bridge the scales and facilitate the bridging of scales from atomistic to coarse grain or continuum via an easy interactive shell. So this project basically would look something like this. The API, you do some import if you have a cloud server, for example, with one available GPU, you just import the libraries you need, you do the installing you need for the analysis. In this case, we're using PICV for the Poisson-Boltzmann part. You import the library, and you can, for example, compare here between SARS-CoV-2 and SARS-CoV-2002 with and without the enzyme, which of it is more stable energetically speaking, in the sense of, and the units are here, kilocal mole. And in the other axis, we can see the pH, for example, the variations in pH and the different types of SARS-CoV spikes. So this project might be extended. That's, of course, the goal of going towards adding genetic material and so on. And we are also open to any collaborations with, if you have a coarse grain force field or a coarse grain method that you would like to add in the project, or some trajectories, it's of course welcome. And of course, the take-home message is that this Pi for All, this free energy characterization, employs just small clusters. It's very democratic in this sense. It's ubiquitous. You can use it wherever you want and from wherever you are. You can also go to feasible models of huge biomolecular assemblies, because you have enabled already some trajectories of other groups of, in the case of SARS-CoV, there are a lot of trajectories of even microseconds of groups in the US that have been shared. And yeah, and then of course another a little bit on the outlook of the projects. Perhaps in this scheme of computer-aided drug design, then also the nanomechanics difference that we found between the SARS-CoV-2 and SARS-CoV-2002 could enter in some kind of these schemes at some point. And finally, the take-home message that multi-scale simulation methods are predicting properties, and are the only method that can allow rapid characterization of biomolecular with a biomolecular resolution. We can also see that it can be used for the co-design of virus-like particles, VLP, and drug design, drug delivery. And of course, the nanomechanical pathways might be of use in the structure-based computer-aided drug design, which might be also a new paradigm for the drug design to add another variable, which is nanomechanics. And well, there is, of course, a lot of space for understanding in the virus world and the virus assembly, disassembly, and so on. And we are just giving here our contribution. And I would like to acknowledge all my collaborators in worldwide, also the institutions where I have carried out this project. And of course, if you have any questions, you would like to contact us for similar projects that we are going on, or to collaborate in the Pi for All project, you can visit this website or just contact us via email.
Okay, so thank you very much, Horacio, for this really nice talk. Just remember that if you want to make any questions, you can just leave the question here in the message in the YouTube. And after the Danilo talk, the two speakers will answer the questions. So once again, thank you, Horacio, for this really nice, really interesting talk. And now I'd like to invite Danilo to give the talk about new strategies for SARS-CoV-2 targets. Just add this to the stream. So Danilo, it's all yours.
Oh, thank you, Rafael, and thank you, Horacio, for your nice talk about the different method to make molecular simulations. This is a picture of Santiago that is just to show one of the consequence of the pandemic issue. Yeah, finally, we can observe the Cordillera de los Andes very clear due that the pollution has dropped a lot. So in that or another context for this talk, I would like to begin to thanking to my collaborator, that is Valeria, Jorle, Romina, Daniela, and Cecilia, that has been working very hard from the beginning of the pandemic event. In that context, is that in our team, we are not just focused in theoretical method, we are also focused in experimental validations. And for that, we have different labs, as you can see, experimental labs that going from microbiology, bio-nanotechnology, biosystem engineering, molecular biophysics, etc., that converge in the same space with the different laboratory that's working with the computational system. For that, we usually begin through the experimental evidence, then taking all the information of this event that we are interested, we evaluate theoretically using different tools, like bioinformatics in general term, molecular simulation, quantum mechanics, depend on the problem that we can front. And then we can take some hypotheses, some questions, even more, that is more usual. It's more usual to find new questions from molecular simulation than new answers for the questions, but it's good. And then taking this hypothesis is validated experimentally. And then we make this cycle several times until obtain new concepts, ideally, and new development for innovation. This is my colleague, that many of them are different specialists in wet lab and theoretical. From in the theoretical point of view, we have in our team Eduardo Castro, who is very well known in Chile, genomics, that is working with bioinformatics, and also Jorle, who is part of our team that is working in chemical synthesis, Ignacio in biophysics, Daniela Aguayo, Egali, Matthew, Jose Manuel, Ignacio, all of them, we are work together in order to solve some problem that we can take all together. In other contexts, we can see that the number of the accumulation of the information in the cyberspace has been increased a lot during the last years. Even more, you can see a representation just in 2015, the relationship between the amount of data obtained from Twitter, astronomy, and YouTube. But the genomic has increased a lot. But that increase is because the technology to obtain sequence has been more cheap during the last years, even more right now, the nanopore technology to the sequence has increased. Include new technology as well has been improved the time to sequence, and the error or the sequences has dropped a lot. But in our case, in the people that is working in structure, we have our own technological revolution. That is the reason why during the last year we have increased a lot the number of the structural information. The main revolution is due that the cryo-EM, single atoms, that you can find a lot of information, even more. The people that is working in cryo-EM has making an incredible contribution to the pandemic research, increases the huge number and very fast the structure of the protein involved in the SARS-CoV-2 virus. So in that context is that for example, Eduardo is working in MetaSub, that is international groups that is taking the genomic of all the bacteria genome of different metro in the world, in the main metro in the world, London, United States, in different continents, and that is making more information for the databases. Even more now, during the next years, probably we have to pass from the terabyte, petabyte to probably to exabyte, zettabyte, and yottabyte. So we have to consider the new concept where is how we can manage our the accumulation of all this information. And also Daniela Aguayo, who is important part of our team, has been working with the different LPS that is part of the bacteria in the surface of the bacteria. It's pretty difficult to characterize. The topology of this kind of system are not being well parameterized. So it's necessary to make long simulation. All that in the context to obtain and try to find new antibiotic. That is the main problem that we also have in the world. As was mentioned by Horacio, that to characterize at atomic level the system that has some interest in health or in technology, whatever, is different. For that, we have a quantum mechanic or simulation of atom, coarse-grain, super coarse-grain as a continuum model. All of them are making different development during the last year. Coarse-grain has improved a lot. Even in South America, we have a very interesting initiative from Uruguay that they are bidding the owner force field for coarse grain. However, the technology required to obtain all this information, as was shown also by Horacio, has increased a lot, and we have more information. We have more information from QIM. Also, will be interested to make simulation even bigger and using multi-scale simulations, as you can see in this diagram. One of the main challenges that we have in our team has been trying to characterize systems that can be observed at a electron microscopy. That is the case with my collaboration with Dr. Ana Maria Cárdenas from CNB, from University of Valparaiso, where we have working during a couple of years with his PhD in order to characterize how a protein can be coordinated around to the lipid layer during the process the endocytosis. Here is how this protein interact with the surface of the membrane, and then beginning the invaginations, constriction, and fission. And for that, this red protein is pretty important. So important that she is very interested in human neuromuscular disorder or disease, just for a single mutation, that is an arginine by a tryptophan. And here you can see the organization of this lipid like a tube, and where the monomer are around to this system. And here it is when we observe these single mutations. Yeah, the question is how, what is the reason of this single mutation can promote so high impact? And for that, Fernando Inostroza, who is the PhD student, built a system first with 2.5 million of atoms that is impossible to organize in this kind of simulation. And then using coarse grain, he built this system with 500,000 beans, and the membrane, to obtain a membrane like a tube, we used a nanotube that was modified in the surface with the heat of the lipid. And then finally, we ran in the system with 56 proteins, a simulation of by 5 microseconds. And then we obtained different characterization how this protein could change his organization. So being to the SARS-CoV-2 in that location, I will just focus in two proteins. One of them is ORF3A, that is a transmembrane protein, not known at the moment, as just a few months ago, we obtained a group from China, obtained the structure of this protein that has not been possible to obtain. Even more, the sequence of this protein is pretty hard to compare with another protein because the similarity is far away to the rest of the protein that already exists in the databases. And also we will talk about a little bit about the spike protein also, but in that case, trying to design antibodies for some specific keys. The experimental evidence of ORF3A is that it's a potassium channel. Here you can see the recently characterization of these channels, that you can see that the classical graph of current versus voltage, and it is in black the current of the potassium, and in red is the sodium, calcium, that is the most important ion that cross through this protein. So apparently, looking this information is a classical potassium channel. Also, when you observe the structure of this protein, look pretty close to a potassium channel with the transmembrane regions and a intracellular region that is it is. It is the nanodisk around to the protein and has a 3 transmembrane member that usually the protein in the virus are very minimalist. So then then this virus can use the small scaffold to make the same function that is observed in another organism. And also you can observe a pore like this and some lateral subunit cavity that can be probably to the interaction with potassium ion through the pore and then cross the membrane. That is basically the observation and the idea that can be observed from the cryogen information. For that, we beginning this collaboration with Dr. Miguel Holmgren and Raul Cachot from NIH and NCI from USA, and with my collaborator, Valeria, Maximiliano, Joel, and Ignacio. We're beginning to try to understand how this kind of channel could be worked. Our previous experience with inside of the Interdisciplinary Center of Neuroscience Valparaiso is that the ion channel that when cross this kind of protein, it generate lines, like can you see? Usually they have a selectivity filter and a cavity, and then it is the extracellular region. In that case, for the topology of the protein is inverse, that the extracellular region is not this kind of loops. It is for inside in the case of the virus, but you can see alpha helix crossing the membrane in order to obtain the membrane. Usually it's observed through the cysteine links, this system. That so you can observe mainly the dimers. Well, the dimer of the dimer of dimers, because each of these subunits are a dimer. However, for us was very impressive to observe that in that case, it doesn't work like an ion channel. Even more, we observe a chloride ion that's going through a lateral windows and go to the inside and is stationated inside, exactly almost in the middle of the membrane of the chloride. So that was pretty difficult to explain to our experimental collaborator, because it's hard to believe that this potassium channel that is observed experimentally as a potassium channel, you can see that it's not a classical potassium channel. Even more, the interaction between these alpha helix is so hard that it's very hard, this interaction, that it's impossible open this alpha helix in order to obtain a channel that cross the membrane. So when we follow working with the molecular dynamic, even more, we observe that until 3 chloride ion can be stationated inside of the main pore of this kind of potassium. And the reason makes quite sense, because inside of this pore, that probably some of the team that is working in this kind of channel doesn't observe, the density of the positive amino acid is quite high. Here you can see a couple of arginine, lysine inside the pore, but also serine, asparagine, among other ions, among other amino acids. So you can observe 3 different chloride inside. After 500 nanoseconds, you can see the density of chloride here in the middle of the pore, and the case of the potassium ion is just in the surface. Yeah, in the surface, because here you can find more negative amino acid. In the case, even more, you can observe a little bit the density of potassium ion in this region. So under this observation in our hand, we're beginning a hard discussion with our colleagues with NIH in order to convince to them that the observation is absolutely different to the basic potassium channel. Here, for example, is when beginning the simulation is pretty positive because here you can find a lot of arginine in the middle of the membrane in the first frame, but until the 500 nanosecond, you can observe that the electropositive density is going down because the chloride can neutralize this position. Therefore, with Valeria, who is in the photograph, we believe that probably the mechanism that is using this kind of channel is not just a unit of potassium channel. It's basically that when the channels are making a dimer of probably a tetramer, they generate in the intramembrane region a positive charge. Therefore, the ion channel don't cross the membrane, no can translocate. That is an important event during the process of the virulations. But when you have the chloride in the middle of the pore, as you can see in green, that is generating a negative electropotential in the middle of these dimers. And then we could observe a huge amount of potassium ion crossing to them. So that this work has been submitted to revision. So we expect to have the full article very soon in some journal. Another case that is pretty important for us is how to develop immunosensor to detect SARS-CoV-2. The problem that we have observed with my colleague Daniel and Romina is that has been pretty difficult to control the pandemic in the world. Due to that, we don't have a vaccine. Even more, we'll have more information about the vaccine. We have the impression that the vaccine can produce a protection for patient by 3 months, more or less. So we will need other solutions, like, for example, have a better control in the detection of the people with COVID. Exists several processes that is possible to obtain the information using the classical PCR that takes some hours. So therefore, it's not the best case to obtain information because even more the PCR tests detect the genome of the virus, but it's reported are slow and require a specialized laboratory. Therefore, we need something more faster. And for that, we are beginning to work in a system that can detect the virus more faster. For that, we are using as the spike protein, that is the most important protein in the surface of the virus, as you can see in the electron microscopy. And also, many group has working like the show, for example, that is his simulation for more than 10 microseconds that you can observe different configuration of them. Well, also that is very important for this kind of protein that interact with the human receptor that is called ACE2, that is here is in red, and interact with the domain of RBD, that is receptor binding domain, that usually are in the upper part of the spike. That is the most important contact with the receptor. And here you can see in orange the sugar that is pretty complicated to evaluate, and even more make more difficult to the immune system to attack this kind of virus because they are mostly protected by sugars. So in that context, we expecting to evaluate different kind of antibody has been appearing during the last month, basically. Here you can see different antibodies that CR30 2020 or BHH72, and this antibody BIT38, that all of them has experimental evidence and also have a structural information. So it's a good point, a starting point to make any evaluation of this antibody. So the idea is to have a nanomolar range KD of this interaction in order to be utilized, in order to be used in diagnostic. But even more, we expecting to improve the interaction between the antigen and the antibody. So if we have the possibility to improve this interaction, our method of diagnostic will be more sensible. So for that, as you can expect it, we can characterize the KD experimentally, but also we can characterize the free energy. And for that, we are using a classical strategy with solvents and using a full molecular simulation of the system. And then we can characterize the contact zones between the RBD and the antibody and measure the interaction using the classical tool that already exists in the molecular simulation field. For that, if we expect them, for example, to change just 4 amino acids in the contact zone, we need to evaluate at least 116,208 different permutations of the amino acid in that regions, because consider that each amino acid has to be mutated for another 19 amino acid in order to evaluate all the possibility that that you have to make in this contact zone. For that, we have in our labs so some computer HPC, the beautiful B100 that also now with the A100 GPU is amazing the amount of the sampling that is possible to do. But using our capability, we can make per year almost more than a little bit more than 1,000 of systems. So it's very limited, our possibility to explore all these permutation between the contact zone between the antigen and the antibody. It is the amino acid that has been to consider some region that is evaluated, and it is the contact zone. And then obviously using different strategy and different a pipeline that has been very complex to implement by Daniel and Romina, is possible to observe in red what are the mutations that has improved the affinity between the antibody and the antigen. But the amount of mutation required, where each system take around to 7 hours per system, I mean, per mutation, 7 hours. So it's hard to obtain. Therefore, if we expecting to make around to 8,000 mutations, for example, we will need to expend around to 6.8 years to make all the simulation. However, with the collaboration with Amazon Web Service, they give us the opportunity to work with them, and then was implemented a pipeline with a lot of detail that can give us the opportunity to make a high throughput screening, making thousands of different simulations using all the computer and facility that they have, and also trying to obtain the best energy evaluation, the free energy evaluation that's possible to do in using these capabilities. Therefore, using our system that 6.8 years that took all this analysis, but using the hybrid HTC HPC architecture, that simulation could take just 2.6 days. So it's an impressionant improvement in the amount of the simulation that can be possible to do. So in around 2 to 3 days, we was possible to make all this mutation that was pretty hard to write the script and the program that is required to make each mutation, and that each mutation has been well implemented in each model. So now we have all this infrastructure that has been implemented in Amazon Web Service. That at the end of our characterization of 3 different antibody, again to RBD, that is the SARS-CoV-2. Finally, we make around 241 microseconds of simulation with more than 6,000 different variants in this 3 system. Okay, so at the end, the storage used for this simulation is 3.6 terabyte. After that, it is the system that has been implemented by Daniel Labs. And the idea is to take some samples and put in this surface is the electrode, which has been implemented with antibody in the surface. Therefore, when it interacts with the virus, that promote an signal, electrochemical signal. Here you can see the work that has been done with the guys beginning with just a computer. Right now, there is the electrode is with graphene, and here is the sample with the potential virus. And then when this take contact with the sample with the virus and the antibody, that produce a signal that has been implemented inside of this box that communicate information with the smartphone through Bluetooth. So here you can see the classical signal of the impedance. So that has been a fantastic work that also has been the collaboration with Cecilia Opaso, making a lot of work with that. And so that has been an impressive experience. I would like to end to thanking again to Daniel and Romina for this great work that finally we are already working in how to produce and purify this antibody and implement in our instrument as soon as possible. Thank you very much.
Rafael? Yeah, thank you, Daniel, for this really, really nice talk. It's really nice. It's really nice to see how we can get this innovation together with the basic science. I really like this.
Yeah, no, me too.
yeah. No, it's a fantastic result. So even my cat like this. She could not get away. Okay, now she's away. So again, thank you very much for both of our speakers with a really, really nice and interesting subject. So now we will start with the questions. We already have one for Horacio from Adolfo Poma, sorry. So Horacio, can you see the question?
Yes. Thank you, Rafael. So the question is, how can we correlate the space of large conformational changes in SARS-CoV-2 and the inner-outer cell environment? I guess in the inner cell environment there are different pHs, and in this case we will have to go back to all atom simulations and do then the free energy characterization to Poisson-Boltzmann method with the exact pH that it's considered in the inner outer cell. And of course, correlating the conformational changes, this is another very good question. Basically, with what Poisson-Boltzmann gives you is a static picture of the free energy. However, you could, for example, use the root mean square fluctuations in some way, and perhaps do several snapshots in time, and then do the whole characterization and when the conformational changes occur, and then evaluate, let's say, the picture from static points, but within a dynamic baseline.
Okay, so thank you very much. Okay, so Sergio just told that they have a couple of questions. Let's just wait while he is probably writing. I have a question for Horacio. It's basically when you go to the post-mortem, obviously you rip off all the water. So I work with water, and my question is how let's say what you lose when you take off the water, because probably water is really important there. So if you go to atomic simulation, you have the water, you have the polarization effects and all this kind of stuff, and probably for instance, the polarization you are losing when you go to the post-Boltzmann. So is this relevant or not? This is my question.
Yes, it is. It is indeed relevant. So the charge regularization, it's not easy in proteins, but when we perform the back mapping procedure, then the simulations are carried out shortly with if we see the case is needed with explicit water, so that we get only the conformation of the proteins, and then through this PROPCA and so on, we will do the characterization of salinity and pH. Yeah.
Thank you. So the first question from Sergio Pantano for Danilo.
Oh, okay. Thank you, Sergio, for your question. So what is the reason for considering the ORF3 as a dimer? Well, the current article in this area that show that the ORF3A can be observed mainly as a dimer because they have between them cysteine bridge bond, but also can be observed that forming tetramer and other superior aggregation of the protein. That is the problem with this protein, that is completely different than anything that you are observed at the moment, at least from my point of view. It's like the virus are from other planets, because nothing is equal to the virus. So for us, it's a surprise to observe this kind of properties during the molecular simulation. Yeah.
Okay. And the second question from Sergio.
Oh, yeah. The chip we are using the impedance. It's a classical electrochemical system that, in that case, in the electrode, we bond the antibody, and then you can observe a specific impedance in the electrode that, in that case, could be using carbon, for example, another mixture of different metals. And then when interact with the virus, that the environment change, and then when it's changed, the impedance, you can observe a differentiation. That reaction is pretty fast. That give us the opportunity, for example, to evaluate the presence of the virus in a sample in just 1 or 3 minutes. Yeah, without consideration that you have to took the sample from the people. So even more, we are trying to make more easy the taking the sample, because at the moment you have to introduce a sampler inside of your mouth. That is it's not very, it's hard to obtain this sample just using saliva. So that will be more easy, that any people can give up the saliva sample, and then you can take information. That At the moment, the system is almost already implemented, and now we are making the first test with real samples, to observe what will be the response of the instrument.
Okay, so thank you, Danilo, for the answer. We don't have any other question in the message from the YouTube. So I want to say thank you again for our speakers. Thank you for everyone that is watching us. And next month we will have another webinar from the SAIMS initiative. So thank you, everyone. Good morning and or good afternoon, depending on the part of the world that you are. So
okay. Thank you.
Thank you, Rafael. Thank you, Danilo.
Thank you,
sorry, Teresa have one question. Let's just wait so she can. Yeah, we have another one. Oh yeah. By the way, for the next month, we have one student from Teresa that will talk. So we have another slot. So if anyone want to give us a talk, just send me an email or send an email to the SAIMS group, so we can organize the next month webinar.
So I have a question for Danilo, actually, very short.
Sure.
And so Danilo, according to this binding energy that you calculated for the different antibodies, which will be the sensitivity you have? I mean, is the method really sensitive, or you have like?
Really good questions. That is hard to find a good method to evaluate the binding energy, even more when you try to compare with the experimental information. But recently, we observed that for example, using BGSA, this kind of method, after 550 nanoseconds of simulation, because also is required to, by each mutation, you need to relax the system. So that in that case, you have to spend some time in GPU to obtain a good sample of this configuration. All the methods is hard to correlate with the KD, but interestingly, we observed that if you use the energy by area that you have connect to the antigen and the antibody, is correlating pretty well. It's a kind of density of energy. Is the density of the regarding what is the amount of energy that you can observe by a nanometer square in the surface contact area. That relationship has been working pretty well, even more comparing three different systems, like the three different antibodies. So that is yes, it's still we need to explore more about what is the best method to. But anyway, we have good candidate to make the mutation right now, and the people is beginning to work in purification of this antibody.
That sounds good. So the error of the method, it's let's say somehow quantifiable as well, or?
At the moment, we didn't quantify specifically the error, but just the correlationship. If you use just the energy obtaining from any free energy calculation binding affinity, it's hard to find the correlation. There is not good correlation. But when you are using the relation energy per area, and then you can observe pretty good correlation. Pretty good is amazing. We need to. We're still exploring how to improve this relationship.
Okay, thank you.
Thank you for your question.
Okay, so now I have a question from Teresa. is a question with a lot of pieces because YouTube, the message on YouTube have just a few number of words that you can type. So it's basically a question about the parameterization of polysaccharides. So here's the first piece. I just put the other pieces.
Yeah, thank you, Teresa, for your comments. That I'm completely agree with you. Well, all right, the Amber, Bromax, so everybody has a good parameterization for the sugars. Yes, it's true. It's true. That the question is that when beginning, doesn't exist this kind of parameterization. We are beginning to work in sugars in the last century, in to 98, for example, we are beginning to work with the sugar. And during this time, it was pretty difficult to find a good parameterization. Even more, in that times, exists some parameterization for sugars that are available for pharmaceutical. You have to buy this kind of parameterization. Even more, Amber has his own parameterization of sugar. But now, 20 years later, you can find a good set of parameterization, and then you can observe this kind of interaction. However, even when you can simulate the LPS pretty well over the surface of the membrane, still you need to evaluate, for example, how phage can interact with the bilayer of the bacteria. And that case, the evaluation of energy interaction is pretty hard to obtain, due mainly because you require a lot of sampling, for this kind of configuration. Well, with the fourth grain LPS, we didn't working with that, and at the moment we are using Romax and Amber to make the simulation with LPS. Yeah. So very interesting.
Yeah. So I hope that is answered, Teresa. Okay. So just concluding, I want to say thank you to everyone once again. I believe that we don't have any other question. I have a few ones, but I will talk with Danilo later, since it's including another colleague that about the. You told that you are using graphene for the. I forget the word, sorry. But later we'll talk in details about this. So thank you again for the speakers. Thank you for the really nice questions. And see you guys in 1 month.
All right. Thank you.
Bye.
Thank you. Bye-bye.
Bye-bye. Bye.